go to, I think, Bio-Edutainment. Bio-Edutainment. Go to the YouTube Bio-Edutainment. So let me post a presentation so that you'll be able to know what you want to do today. Okay. Are you there? Bio-Edutainment. Do you see anything upcoming? You should be able to hear me now clearly on Bio-Edutainment. You already have this. Check if you can see my screen. Not yet. You should be able to see this. Who has seen it? DIY, Bioinformatics DIY. If not, oh, sorry. There are certain things I was supposed to have done. Can you hear my voice? Hold it now. Maybe to this. Huh? Yeah, well, okay. So that is what we want to do very briefly. Don't forget the last class. The last class I told you hello. So the last class, can you hear my voice? My voice is not coming up. Not coming up at all. My voice. Is it coming or it's not coming? Behind herself. Let me see. Okay. You should know that you don't expect it to be at the same pace, to be moving at the same pace. But I'm sure it's not on echo. The voice is it on echo. The last um, the last class we had, the audio was so bad that nobody talked. The audio was so bad because it was on echo. If you later go to that video and you watch it, you'll be able to see that it is on echo. So just like I said, what we want to do last week or last lecture, that should be on Monday, I think. So what we did, by now, you should be able to know how to blast, the logical reasons or the reasons behind blasting. You should be able to also know what e-value stand for. You should be able to know, even from there, how can you differentiate between prokaryotic and eukaryotic. Also in that presentation, I also told you about the trinity, the DNA to RNA and to protein. And I told you, I wanted to make an emphasis. I told you, if you check the WhatsApp group, I sent you the 20 amino acids yesterday. That 20 amino acid is something you need to know, you need to learn, because those 20 amino acids are those things that will form a single protein, I told you. So having said that, I also told you the differences between multiple alignments and pairwise alignment. And I also told you how how do computer interpret biological information, especially when it comes to matching them together. So I told you what plus one stand for, what minus one stand for, and what plus two also stand for. Then I also emphasized on the biological implication of um, the replacing, maybe a single nucleotide being replaced, you know that it's going to get into what is called mutation. And I remember I also cited an example, a typical example of where mutation takes place. I gave you two examples. I mentioned the sickle cell anemia, and also mentioned the case of drug-resistant tuberculosis. I also emphasize on that. Not only that, I also gave you a classwork, and that classwork you are able to retrieve by yourself. You are able to retrieve certain information from NCBI. Just typing the name of any organism of your interest on NCBI, and you are able to see what actually came up. And I told you that as a bioinformatician, the code or the format that is acceptable to be used in bioinformatics is known as FASTA formats. I gave you an example. I said you should type in the object, the organism of your interest in which you did, and I told you to generate it in faster format, whereby I said you should click on the first five or any five of your choice from the gene bank. So what I will also tell you is just that what are the differences between there's a place when you get to NCBI, there's a place if you click on um, if you click on blast, there are section of blast N where you see blast P and T, blast N, and so on. We will see it and you see what it is actually standing for. So without wasting our time, I think your background is already getting more robust, and by now you should be able to do certain basic things by yourself. Now, this is just a kind of a furtherance lecture, and that is why I call it bioinformatics, which is DIY. That is do it yourself. Do it yourself. Monica. So DIY means do it yourself. So that is, even when there's no internet, you should be able to do all this on your own. You don't need anybody to tell you. You don't need anybody to teach you any longer. So what you actually need is this. So that is why what we want to do today. And looking at what we have on the screen, don't forget that I told you all information, anything that has to do with bioinformatics must start with what is called the central dogma theory, which I call the flow, genetic flow of information from DNA down to RNA, then down to protein or amino acid. And I made emphasis in the last lecture as well that the structure of protein determines the function. That is, anytime there is a kind of misfolding in the structure of protein, it is going to go a long way as affecting the general function it is going to perform in that organism. So what I'm trying to let you know so far is that you should start your knowledge from the DNA. So DNA, and by now you know that DNA is made up of 4 nucleotide bases, which are adenine, guanine, cytosine, and thymine. It is in RNA that you have the T being replaced by U, that is uracil. So even if you see any sequence now, under normal circumstances, you should be able to differentiate between sequences that are coming directly from DNA and sequences that are from RNA. Then, by now, you should be able to know and work. In today's class, we are going to also work, perform blast using using protein, that is amino acid, which are the building blocks of protein. So without wasting our time, we want to analyze the real biological data without writing a single line of code. You don't need Python. You don't need any computer knowledge before you'll be able to analyze all this. So just like I said, you as a bioinformatician, these are part of what you should be doing every day. And I told you in the last class as well that when we talk of diagnostic or diagnosis, so you can easily use, once you have your organism, if you have it in form of code, it will be very, very easy for you. Since you have that code, get your code, and which we call the query sequence. You get your code, go and compare it with subject subject sequence in the database. So, and anyone that has almost 100%, or where we have the e-value tending towards 0.001, that is going to be more significant. So, having said that, today, just like I said, let us begin. Biology is now a data science. I've told you several times that gone were those days that you have to, as a biologist, what you know is you just want to make use of a microscope. You just want to be wearing lab coats around. What you just, if you say, one of the reasons why some people feel about to be more important than others is because of the courses they are studying. Some will tell us that environmental biology is more inferior than microbiology. Some will tell you that biochemistry is much more global and acceptable than any branch of biology. So, and that was then. If we are talking about bioinformatics, gone were those days that you have to cram all the cellular pathways, cram Krebs cycle, glycolysis, nucleic acid synthesis. You cram all this one, and at the end of the day, what you will do is that you will still go and forget, just for you to learn it in the exam and pass the exam. After that, you won't have any other thing to be able to do. But I just like I told you, bioinformatics is what everybody is talking about now. So biology now is now a data science, whether you like it or not. Uh, whether you like it or not, it is now a data science. So historically, limited by manual observation and physical tools. In those days, when we used to use a microscope, you just discover that you just continue. In fact, there are some people that up to today, they cannot even work efficiently on microscope because they don't know how to operate it. Even if they know how to operate it, what they will be looking for under the microscope, except they are trained, they might not even know that they are already missing out essential things. So that was then. That was then. That was the era where misdiagnosis was very, very possible, where mistreatment was very, very possible. But nowadays, that biology is being worked upon in codes. There's no more misdiagnosis. I told you that one of the major gain of bioinformatics is that we are working towards personalized medicine, personalized prescription, personalized diagnosis. The old diagnosis that will only be meant for you and be meant for you only. For example, it is very, very possible genetically that you can have the same species of organism. You can have E. coli from your patient A and the same E. coli from patient B. In fact, don't let me even talk about two patients. Let me talk about even you. You can isolate a bacteria from your left ear, the same bacteria in your right ear. They will have this, different sequences. They will have, that means they will also require different treatments because they have different, on the same individual, you have the same species but of two different sequences. So what we are saying in essence is, if your ear is having problem, you isolate an organism for your ear, you should be able to tailor the diagnosis based on the left ear, and you should be able to also tailor it based on the prevailing environmental condition. So that is exactly what we are saying, that bioinformatics has actually giving us a lot of gain. Gain in the sense that as a result of it now, we can talk about personalized medicine, personalized prescription, and its applicability in other fields. So we are looking at the traditional, the biology of those days. We are looking at it. We are looking at biology of then. We are looking at it as versus the biology of now. Now, don't forget that DNA, the biology of today is biology of code, biology of letters, biology of data, biology which is called of information. And I told you that DNA, RNA, and protein, they are called the informational molecule. The informational molecule. Because when you combine all these letters to you, it has its own meaning and pronunciation in cellular world. So bioinformatics uses massive computing power to decode it faster than human. So I told you that in those days we now have a lot of information, data that is being generated every seconds. In fact, if you go to gene bank where data is being deposited, that gene bank, you you have it you have data being generated every minute. the number of the data that was there last week will be quite different from will be more than what you will see. So information are being generated every day. So that even if you say, in fact, you see one thing, when I was trying to do analysis of one of my work, just for one information, when I did the first analysis, I had 800 pages 800 pages of just one. So how am I going to comprehend and interpret this and make meaning out of just one just one sequence like that? So, but we need computer to be able to process this. So that's why you said that the nowadays biology, which is the biology of DNA, the biology of DNA is making use of data, is making use of code. It is so massive that it will require computer, the head of computer, to be able to interpret it and even analyze it. Now, let us now face the reality. The reality now is today digital lab protocol. Today is you see, in those days, you see WHO building very big, gigantic building to have sophisticated equipment. That they would now spend so much money to hire personnel that will be able to even man it, be able to operate it, and so on and so forth. But nowadays, they don't need it any longer. What they are looking for is they are looking for bioinformatician. So do you know that even in your room, your own room, you can have your own data lab there. Once you have your phone or you have your laptop, you have source of internet, and you have access to the data bank or the gene bank, you're already a bioinformatician. They can be consulting you from there. Please, we want you to analyze this data for us, and they will do it for you. You will do it for them. So, the today's digital lab protocol, number one, is you should be able to have access a real scientific database. By now, you should be able to know if you are having, depending on the information you are having. If you are having information that has to do with DNA, you know the database to go. If your information is on RNA, you know where your database should be. If your your information will require protein, you know where to go. So the first thing is how can you access a real scientific database? So the second one is that you should be able to run a protein sequence analysis. Under normal circumstances, you should be able, by what we will learn today and what we have been learning so far, you should be able, at least by now, one of the basic things that you should be able to know, do, even without help of anybody, and that is why we call it DIY. You, as a bioinformatician, can you do it? So you should be able to run a protein sequence analysis. So if you have a sequence, by now, you should be able to compare, do blasting, do blast about that little sequence that you have. Then you should be able to decode the results like a bioinformatician. That is, you should be able to interpret. You should be able to make use of it. Not that you just and you don't have to stress yourself. It is computer that will do the analysis for you. So, and what are the prerequisites? The prerequisite is just your web browser. Once you have your internet and your cell phone, fine, you can practice bioinformatics at any point in time. So that is the biology of today. Now, just like I said, your bioinformatics toolkit. So you know that you cannot. How will you see? Okay, for example, if somebody is a mechanic now, and the person doesn't have a tool, how will you? Can you be so? Can you be so confident of taking your car for the person to go and repair it? Not possible. It's not possible. So, as a bioinformatician, what are the toolkits that you must have? the basic tool, the basic tool that is Edu. For example, now somebody is a fashion designer and the person doesn't have scissors. The person doesn't have a machine, which is supposed to be part of the tool. But irrespective, if somebody is now telling you that, yes, I am a global international fashion designer, but I'm not making use of, I don't use, I don't use machine, sewing machine. Me, I don't have scissors. Me, I don't. Will you be so comfortable with that person? No. The answer is no. So what we are saying in essence is, as a bioinformatician, the necessary kit, the necessary tool that you must be able to know, you must be able to use at all. Basically, you should be able to use NCBI. Let NCBI be your guiding angel. Let it be your consultant every time. So, is the database the ultimate library of all biological data? So, any information that you are looking for that has to do with biological data, you should be able to know what NCBI will do about that. And when you get to the NCBI, it's like now you now have a tester as your toolkit. And at the end of the day, you just have it, but you don't know how to use it. So that is why when you get to NCBI, you should be able to do what is called a blast. So, and what is blast? Blast is the search engine. I told you, the algorithm used to find matches inside that library. So what I'm saying in essence is, at least by now, if you don't know anything at all, nothing at all, you should be able to know how to blast, how to solve the internet, and how to pass the internet to be able to retrieve sequences. And when you now get to the NCBI, NCBI is not only for DNA, I told you. It is also where you can get information about RNA and you can get information about DNA and protein. So the only thing is that it is not a structural database. It is not where you will get the structure. It's not where you know. For example, now you now have a long thread of amino acid that is going to form protein, and you want to know the structure. Don't forget that I told you once you are working with protein, what should be coming to your head should be that this protein, is it operating at primary level, the structure? Is it at primary level? Is it at secondary level, tertiary level, and quaternary level? All those are very, very important. Hemoglobin, our normal hemoglobin now, the difference between when you are considering circulation, we have deoxyhemoglobin, and we also have oxyl hemoglobin. Deoxy is when oxygen is already being removed, and that is telling you that it is going to be systemic type of circulation. So what I'm trying to tell you is that you, as a bioinformatician, should be able to do and perform the task like that. Now the next thing is configuring the sequence scanner. Configuring the sequence scanner. And this is going to be a special focus on protein. It's going to be a special focus on where? Protein. So, and what will you do and what are you expected to do? You have to navigate to NCBI. When you get to NCBI, you will go and select blast. Click on blast. When you click on blast, go and click on blast P, because that P is standing for protein. Blast N is standing for nucleotide. So we will see it when we are going there. So why proteins? Protein sequences are easier for beginners and yield crystal clear alignment results. Now, under normal circumstance, I want you to know that 3 letters code for one amino acid. 3 letters code for one amino acid. So that is telling you that if you have 300 nucleotides, 300 nucleotides, that is to tell you that how many amino acids do you have? Divided 100. Good. 100. So it is easier for you as a beginner to always want to work with protein, amino acid, than working with DNA, because the DNA is going to be too enormous. Look at the whole genome sequences I asked you to download the other time. That I think somebody was trying to download it in her phone, and the memory could not even contain it. So because the data was just too high, too many, too enormous. But if you are working with protein, that means the sequence you are working in is going to be divided by 3, and that is can actually tell you. So it is easier to work with protein than working with DNA. Now, I want you to take note of this. Please write this one down. You will see it in the on the screen. You will see it on the screen. Okay, loading the biological inputs. Now, don't forget that the first one you have gone to NCBI. After NCBI, you now click on protein blasts. Okay, when you now click on protein blast, you now enter this information in that. Okay, is that page already, the screen, is it displaying to you? MVLS, abi? Okay, so you now enter it into that space. So this is a small known protein sequence specifically used for learning. So for a beginner like you just start from here. How many amino acids do you now have there? How many amino acids? 1, 2, 3, 4, 5, 6. That is 12. M is standing for something, V is standing for something, L is standing for amino acid, but all these 12 we have its own interpretation. So, and when you want to understand amino acids, there are 3-letter codes for amino acid. There are single letter codes for amino acid as well. So I'm very sure that some of you must have learned this in your days of cell biology. Now, executing the cross-referencing. So when you click on the blast, you will see that the system is concurrently comparing your 12-letter sequence against millions of recorded sequences across all known life on Earth. So immediately you now click on blast, after you must have entered the 12 amino acids. Once you click the blast, your blast is going to run. And what is it going to run? Is comparing your own sequence with millions of sequences known throughout not only in the world. What's the difference between world and the heart? World and heart. Which one is bigger? Okay, you can find out. But what we are saying in essence is just that the heart is one of the planets now. That's your elementary geography. One of the, I think, 7 planets, Abi. And now they said we now have 8. Don't know. I'm not a geographer. Okay, so that is that. Okay, now what you now see is you now see a result that will be displayed. Hello, please do that. I want you to do what is being displayed. what I asked you to do. Go to NCBI. Or you want me to finish the presentation? Okay. So when you now anatomy of your blast results, when you after you must have blasted your results, this your sequence matches some proteins. So you are to ignore the noise and focus on just these three. Only three values are going to be very, very important for you. Number one, the organism. Number two, the score. Then number three, the e-value. Organism, score, and e-value. So for me, I will also ask you to put number one. Another one is accession numbers. You have to put the accession numbers because sometimes you might not even need all the sequence to input all the sequence. Once you enter correct accession number, it's going to display the sequence for you. So please, you have to know it. So, and if you are working with if you are working with protein, the first one, is your protein. Is your protein query sequence. Don't forget, okay? So that is the analysis. Now, decoding the e-value metrics. I've told you the e-value is the most critical, the most important. In fact, number one thing that you need to know about running a blast is the e-value. In fact, sometimes it is the e-value that will tell you how identical are they, how related are they evolutionarily. You know, I said this in my last class. So it calculates the statistical likelihood of this genetic match happening purely by chance. So e-value, what e-value does is that the e-value will now compare and tell you what are the chances of what of random occurrence. Okay, and when you now look at that scale, you see a scale where you have I think this color is orange, abi, the one towards the left, is that orange? Okay, that color towards tending towards orange, that is higher number and random coincidence. But when it is turning towards that green, that means it is approaching perfect match. Perfect match. So what we are saying invariably is that if you have smaller number, that is a better chance of matching. Once you have a smaller number, that is telling you that the match is going to be almost perfect. Okay. Now, don't forget, you have a sequence being displayed to you. So in less than 2 minutes, we want you to be able to track. You want to track when you have that protein. That protein is just coming from an outbreak. Just the same thing they did to COVID-19. They just got hold of the protein. They wanted to know which family it belongs to. They wanted to know how can they diagnose it. They wanted to know what are the treatments, the best treatments they could give to it. Okay, and what are even the symptoms. So don't forget, the sequence are being gotten. The sequences, nucleotide sequence was was retrieved from the genes. Can somebody tell me the difference between a gene and DNA? Gene and DNA. Okay, a gene is segments of DNA. Segment of DNA is a gene. So a gene can just be 20 amino acids. It can just be 30 nucleotide sequence, but it is located on the DNA. Okay, so that gene you have, this one was traced back to COVID-19. Let us see whether it is that. So it is from here, as you have that protein on displayed on the screen, that is operating at primary structure of that protein, because it is still on straight line. Amino acid on straight line. It is what? The primary structure. And by the time they now perform more structural prediction on it, they now discover the real structure. So the structure here now is at a secondary level, where you have beta and alpha together. I showed you some of the how to identify some of these in the other one. So the mini challenge. So we just we want you to simulate, we want you to predict a kind of a mutation. And what do we call mutation? Mutation is a sudden change. It's a sudden change in a genetic makeup. So when you have the original sequence, we want you to also predict mutated sequence. Apart from what you have here, so let there be another mutated. You can also put your own to have in another place. So instead of this, but what we have on the board is we just have a point mutation where you have A, it is now being replaced by E. So scientific exploration require asking what if. What if? The what if is that what of if there is any change in the original sequence? When you run the blast, is it going to be the same again? So those are the challenges. So you have to your whole you don't have to simulate the same in the same way where A is being replaced by E. Just assume that your point mutation occur somewhere, because you are going to use different amino acid to run your blast. So I also want you to run blast to see what is going to happen. Are you going to get the same E value? Are you going to get the same number of match if you are dealing with mutated sequence? So does this resulting organism change from the original one? So this is one of the reasons and one of the significance of what is called mutation. And that is the reason why it is very important for you to be able. Don't forget, I told you that you should be able to start your bioinformatics journey with preferring to make use of um, protein, amino acid, rather than starting your journey with the DNA, which is a nucleotide. So, and the bioinformatics skill tree that you have to know, or you have to at elementary level. I told you there will be a time. I think I told you sometimes ago that you should begin to first semester. I'm very sure that you should register for Python free of charge. So people started when I said there was going to be 30 days Python challenge. People registered and never showed up. Anyway, that is not it. That is not the cause of this, because then I told you why. One of the reasons that you needed to register for it was free. Now I don't know. You can still have a free training online. I don't know. Not even from me, maybe from other days, but even after some time now, I will start the class again, and not minding whether you join or not. I'll just start the class again so that anytime you can visit the website and see. So the future of biology is digital now. So nobody is talking about your knowledge about microscope any longer. So biology is no longer confined to the petri dish. It is driven by data. It is scaling up by technology, and you are only limited. Do you know what is only limiting you? Huh? Do you know what is limiting you? What is limiting you is not your ability to use data. It's not even that you don't have money. It's not that you don't even have access, that you don't have equipment. When you are saying, man, because I don't have laptop. Yeah, your phone, call your cell phone. In fact, there are some of your cell phone that are even more expensive than some laptop. So you don't have any excuse. The only excuse you can have is your curiosity. Are you ready to learn? Are you prepared for the world to come? I think last year I gave a kind of somewhere I was invited to, whereby I gave a talk on um, bioinformatics, the science of 2055 and beyond. 2055 and beyond. So that is to tell you it is the science of the future. Many of us might not be there any longer, but those that will be there, if they do not have this knowledge now, they will not be relevant. I remember maybe there was a time very shortly, I also talk about AI is not coming to replace you, but people that knows AI will come and replace you. Huh? If you refuse, it's not AI, because AI, you are the one that is going to prompt AI to do something for you. But if you don't know how to make use of that AI, honestly speaking, people that knows how to make use of it, you can see many people are making money even with ChatGPT. Many people, ChatGPT, which is very common, that you can even have free, you can have them free on internet. I told you the other time when I was talking about innovation, moving from um, dreamer to innovation level. So under normal circumstances, even if you don't know anything at all, just gather PS3 students together. No ChatGPT. Okay, you want to teach them circulation. Just, oh, I want to teach a beginner circulation. It's going to give you everything that you need. Going to give it to you. And before you know, even if you want to diagram, you don't need to start drawing diagram for you. Just show you them, teach them, whatever. Mathematics, yeah, I don't know how mathematics is. You, when you know how to use ChatGPT, no subjects. Let me tell you that no subject is going to be too difficult for you. It's going to be too difficult for you. The EduInteractAI, my ecosystem, I told you that at a point in time, in the next few days or few months, when you just visit the website, there will be a time that it is the AI that will be welcoming you. You'll be talking on that website, you'll be talking to that agent that, please, I want to learn bioinformatics. Okay, if you want to learn bioinformatics, these are what you need to do this and do this and do that. So the world over, is no longer limited, biology is no longer limited to petri dish. It's no longer limited to, I want to culture. I don't know how to carry out a gram staining. I don't know how to carry out other staining. Apart from that, you also expose yourself to a lot of risk. So the biology of today is the biology of data. And that is why we are calling on you. That is one of the reasons. I think the last class, I just told you that you should just try and look for what you mean by EduInteract AI on the um, which AI did you use? Which AI did you use that day to search for it? Was it Claude or what? Or Meta? Which one did you use? Was it on Meta? So to actually tell you whether you are in the right place or you are not in the right place, what you have access to, the opportunity that you people you have today, having something like this around, so many people did not have it in the year past. And that's the reason why I will want you to continue, just like I told you, continue with that. A time is coming very soon. Just open that website. Just prompt the website. Teach me. I want you to teach me. You can even tell the AI, teach me like Mr. DOT will teach. It's going to tell you, it's going to start teaching you. Huh? It's going to start teaching you. My voice, my everything is going to be there because it's already been simulated. So even if you say, okay, please teach me, help me predict what will be the likely question on this subject, it's going to generate it for you. Gone were those days that you will just say that, me, I don't know what to read. Your problem is no longer you don't know what to read. Huh? Your problem is that you are too lazy. You are too lazy to do what you're supposed to do. Up till now, there are some of you now that even you are not yet subscribed. You've not even subscribed. And where are you going to get? It's only when you want to, when there's lecture, that's when you just join. So what we are saying is just like there are other things that you can gain from there, not only on the that. Hey. So your laboratory is open, already open, already.